Okay, the main difference is going to be ATP. If ATP is going to be being used, then it's going to be active. If it's not being used, then it's passive. Okay? And so there's going to be two types of passive transport that we're going to talk about. So there is facilitated diffusion, passive diffusion. Okay. So what was diffusion?
Diffusion was it was like, didn't like the absorption of I can't remember.
Super, super, super important principle. Diffusion, the basic idea behind diffusion is going to be things diffuse from high concentrations to low concentrations. If I put a cookie, fresh baked cookies, put it on the counter, particles break down, right? And they diffuse throughout the house, and then you can smell them. So that high concentration cookie breaks down and it diffuses to low concentration. If you left the cookie there for like 50 years, then it would break down all the way and be diffused everywhere. Granted, you'd have to leave it there for a long time. But some other things are in play in cookies. But so if there's a lot of sodium outside of the cell, if it can get in, it wants to flow in, right? If there's a lot of potassium inside the cell, if there's a way for it to get out, it wants to flow out, right? So that's the principle of diffusion. Super important principle. Do not forget it. Okay? So that's diffusion. Okay? Some things can freely diffuse through the phospholipid bilayer. Do you remember what they are? What things can just go straight through?
I don't remember.
It's going to be hydrophobic or hydrophilic things. Okay, so heads are what?
Hydrophilic.
Hydrophilic. Tails are hydrophobic. So I told you how these things are moving all around, right? They move back and forth, changing spots and whatnot. So something that is hydrophobic, when it's coming along, it can get past the small areas of the heads pretty easily. And then once it's in the tail area, it does just fine, because these are hydrophobic too. So it can move through them just fine. And so it can go straight into the cell. Pretty easy. But something that's hydrophilic needs some help getting through. That's why there's carrier proteins. Okay? So the carrier proteins help it so things that are hydrophilic can get through. Because the hydrophobic things can get through by themselves. So passive diffusion is going to be for things that it's a very passive process. It just diffuses through. Whereas facilitated has to have something to facilitate its diffusion, like the proteins. So hydrophobic things are going to do passive diffusion, whereas hydrophilic things are going to do facilitated diffusion. Okay? Facilitated diffusion can either be so leak channels. You talked about those before, or looked at them in the book, right? Okay, so leak channels. There are group all of them together. Gated channels. Like voltage gated and ligand gated channels, right? And I think those are the only ones that you have to talk about. But yeah. Okay. Is that all right?
Mm-hmm.
Active transport needs energy. So why would you need energy? Why would you have to use energy? Any ideas?
The only thing I can think of is to like give it a boost.
Yeah, okay, it's giving it a boost. Diffusion, high concentration to low concentration. Piece of cake, it just does it by itself. But if we want to pump from low concentration to high concentration, that's going to take energy. Okay? So active transport, there's two categories. There's primary and secondary. So primary active transport and secondary active transport. Okay? The primary are going to be ATP powered pumps. The biggest one that you'll see of this is going to be the sodium-potassium pump. Because, so in a cell, just as an FYI, there's a lot of potassium typically inside the cell and a lot of sodium outside the cell. So there's this sodium-potassium primary active transport pump that pumps sodium out, potassium in. So it takes energy to do that because it can't just diffuse down its concentration gradient. Okay? Secondary active transport doesn't use ATP itself, but it needs an ATP to work. For example, so calcium is high outside the cell. Okay? So what this does, it's another antiport, anti being against or opposite directions. So there's another antiport pump which lets sodium go in, which is from high to low concentration, right? So diffusion, no energy. But then pumps calcium out. Okay? From low to high concentration. It doesn't use any ATP itself. It's powered by this concentration. Because that being able to diffuse down its concentration gradient it's releasing energy, you know? Kind of like water. Here's water, here's a stream, and here's a power wheel. You know, one of those things that's spun by the water. So it's using the energy from the water flowing down the hill to spin it. It's using energy from the sodium flowing down its concentration gradient to pump the calcium out. Okay? So secondary don't directly use ATP. Okay? So real quick, ATP powered pumps. That was one of your questions. Did I answer that? Okay. So in a lot of these things, sometimes this is used to send signals or to make something happen within the cell. Okay? Like we might open a calcium gate to make our muscles flex because calcium's how that's powered. And so it's causing something to happen. To save drawing time, I'm going to use half of this picture. Okay. So this is going to be the cell membrane. Okay. Yeah. Okay. So for the G proteins, so the G protein complex is what it's called. We've got a couple parts. We've got the G protein complex right here, and then we've got a membrane-bound G protein receptor. Okay, so this protein stays here, is bound to the membrane, has a receptor up here. Okay, so when a ligand is basically just going to be particles. So a specific particle, like one particle name is acetylcholine. Okay, so if that comes, binds here, it wants to send a signal or to cause something to happen. That's our purpose. So this guy, the G protein complex, it comes over to him and it gets energized. Okay? So this guy, instead of ATP, it has a different energy source. So it's GDP. So remember how there's like ATCG? That's the G from that. So guanosine diphosphate instead of adenosine diphosphate. So that's attached to this guy. He gets charged, so it goes from GDP to GTP. And then he goes and sends the message. Okay? And that's all you need to know. Because there's a million types of G proteins that all do different things. But this guy goes and sends the message. More specifically, the alpha subunit, because there's 3 subunits: alpha, beta, and gamma. Alpha, beta, gamma. Beta. Okay? So basic idea, receptor, this guy comes over, gets charged, and he goes and sends the signal. Okay? So that's what the G proteins are. Si bueno? Si bueno. Cool. Okay. How you feeling? What are you thinking?
It makes a lot more sense.
Yeah.
I feel good about it.
Because instead of just saying ATP powered pumps and the G proteins, I thought if I kind of break it down, then it will help organize some thoughts. What was I hoping to do? Did it help?
Yeah, it did.
Okay.
I love